Okay, thank you very much. I would like to thank the organizing committee for giving me the opportunity to present some data of a larger study onto the subject of autism and ADHD, and especially with relationship to substance use disorders. And the data I'll be presenting here was recently published in the World Journal of Biological Psychiatry, and the co-authors are listed on the slide. So the background to the study is that in clinical practice, autism and ADHD are often difficult to differentiate, especially when there are comorbid disorders. In our Department of Addiction Psychiatry, we noticed that some patients with serious substance use disorders on closer examination appear to have autism, and that had not been diagnosed before. Instead, the condition ADHD had often been erroneously diagnosed in these patients. Now, because there was no literature on the subject of comorbidity in substance use disorder and autism, we set out to conduct a study, an explorative study, on a broad domain, several domains, parts of which have been published elsewhere. And here, as said, I present the findings on the analysis of 5 candidate genes in adult patients with autism or ADHD with or without substance use disorder. Now, the heritability of autism and ADHD and substance use disorders is quite high, suggesting a large genetic contribution in the etiology of these disorders. And, for example, Angelica Ronald published twin data indicating that there was a significant overlap between patients with ADHD and autism in the general population, with correlation factors more than 0.5. Also, a recent publication, of course, in The Lancet by Williams and colleagues showed that ADHD is characterized by large and rare CNVs that show overlap also with autism and schizophrenia. Now, several studies have looked into shared candidate genes for ADHD and autism, but our interest was whether candidate genes could differentiate, especially in the condition of comorbid substance use disorder. So we examined 110 normally intelligent adults, 61 with autism and 49 with ADHD. And of these, 43 people with autism did not have a substance use disorder, and 21 of the people with ADHD did not have a substance use disorder. We genotyped for polymorphisms in 5 candidate genes, which you see here below as 10 groups. We genotyped for the dopamine D2 receptor gene, often associated with ADHD. We genotyped for a contactin-associated neuronal cell adhesion gene, CNT-NAP2, linked to autism. A neuronal cell adhesion gene, CDH13, implicated in ADHD and substance use disorder. And fourth, we genotyped for the rate-limiting gene in the serotonin synthesis, TPH2, related to ADHD. And the serotonin, last but not least, the serotonin transporter gene, whose polymorphisms are related to the speed of serotonin metabolism in autism, but is also implicated in ADHD and in substance use disorder. So what we did is we grouped the allelic variants for each gene into 2 groups, indicating the presence or the absence of an allele, which you see here. So for example, for the serotonin transporter, we have a group for the short allele and a group for the long allele, and the group for the long allele contains both LL and LS, and that's how it works. So we obtained 10 groups. So we carried out logistic regression analysis models for each of these 10 groups, with the allele group as the independent factor, and the diagnosis, substance use disorder, and the sex as the independent factors. Now, what we found is that 3 significant effects for diagnosis. In the first place, we found that the presence of the CNT-NAP2T allele, which you see in the green bar, predicted ADHD rather than autism, with an odds ratio of 2.67. We also found that the presence of the TPH2G allele, which is indicated by the blue bar, and the long form of the serotonin transporter, the yellow bar, predicted autism instead of ADHD. But mind you, this was one group with either ADHD or autism, so it's not in the general population. And there was no effect for lifetime substance use disorder, so that indicates that if there is a clinical implication to these statistical relevances, it would mean that if you have to decide between ADHD and autism, and you know it's either of those two, it could have clinical implication, and if there's also a comorbid substance use disorder, it would not affect the differentiating ability of these candidate genes. So the results suggest that two serotonergic genes and a neuronal cell adhesion gene can differentiate between autism and ADHD patients, and that substance use disorder does not affect this ability. However, as you've seen, the numbers are very small, and although the statistical analysis is robust, this calls for replication in other samples with larger numbers. Thank you very much for your attention.